we will move on to the production of the protease. So in production of protease, we already know that there are upstream and downstream processing. In upstream processing, we have to be very specific about the raw materials, the microorganism used for the production of our desired product. And then we will frame it down, the seed fermenter and also the production fermenter, the optimum conditions for each and every microorganism. Either we are going to use it for a normal either organism or a mutant strain. Based upon that only the overall production can be increased and the yield will also be very good. So while conforming all the microorganism which is commercially available or which can be easily produced, the high yield can be obtained, that organism will be specifically identified and we will go for the production. So when we talk about the microorganism, these are the microorganisms which can able to produce this protease enzyme, and we know that there are different classification, and based upon that classification also we can say like these microorganisms will also vary for. So first one is Rhizopus microsporus and B.licheniformis and B.amyloliquefaciens and B.firmus, B.megaterium, B.subtilis, Streptomyces griseus, and some of also the fungi will also plays an important role for the production of this protease, like Aspergillus niger, Aspergillus flavus, Aspergillus oryzae. So all these microorganisms can able to produce the industrial protease enzyme, and it requires a pH of 7 and the temperature will also vary from 30 to 40 degrees centigrade. So this is what the overall, first we will first frame it down the microorganism, the inoculum preparation we will do it, and then we will move on to the seed fermenter, same like whereas we have only one particular microorganism here. So the overall enzyme production, maybe it will be like a submerged fermentation process or a solid substrate fermentation process. Already we know that what is submerged and what is a solid substrate. So if we are going to use only the broth as a substrate, all the nutrients will be mixed along with the distilled water, that is called submerged fermentation. So solid state fermentation is that substrate itself will be a solid material through which we will spray the microorganism, and also if the processing, the production of our product will be happen inside that solid, we will separate that substrate as well as the solid substrate as well as the product, and we will undergo the further extraction process and all. So this is what the difference between. So the microorganism may require sometimes a submerged, or as if some of the microorganism may take the solid substrate fermentation. So if it is in submerged fermentation, we will directly go for the cell separation, and then we will separate the biomass, which is nothing but a microorganism, and then we will collect the supernatant. We will undergo the enzyme concentration of it, and then stabilizing of that product. Then the enzyme product, purified enzyme product, will be obtained. So this is the overall thing about the submerged. So when we talk about the solid state fermentation process, after the solid substrate, we will extract on adding some of the solvents. We will extract only the product from that, and then the solid, whatever the solids utilized by the microorganism will be sent as an animal feed. This will undergo decomposition, or else these animal feed will also, the solid waste will also be decomposed, and the production of biogas can also be occur, and this biogas can be utilized for the overall plant. And if the action is over, then we will go for the next step, that is cell-free supernatant will be again treated over. Then the enzyme concentration, stabilization, as well as the enzyme product will be obtained. So this is what the overall production in an industries we will go for this production of this protease. Then we will see that only one specific microorganism, Rhizopus microporous, will be used for. This requires at least 5 days for the recovery or regeneration of that particular microorganism. So once it have been desuspended, if the pore have been formed till 10 power 5 of for ml itself. Then we will move on to the submerged fermentation. So we are going to look for only the submerged. So in submerged fermentation, we know that the media plays an important role. So whatever may be the media, it requires the salts or else nitrogen sources, carbon sources. All these things will be framed, and we will go for the sterilization process. Then only we will inoculate into our seed fermenter. So the media composition required is ammonium sulfate, then cobalt diammonium hydrogen, then potassium dihydrogen phosphate, dipotassium hydrogen phosphate, calcium chloride, and magnesium sulfate, iron sulfate, manganese sulfate, zinc chloride, and the nitrogen sources such as peptone, yeast will be used, and some of the substrate as in rice bran have been used. So this is the media composition, and based upon this we will also use some of the molasses for as a carbon sources, and in some of the industries they will also go for the beet molasses, sugarcane molasses and all. Then once it have been over, the overall upstream processing we are going to see it. So rice bran have been added, and it is autoclave. It have been inoculated, the spores have been inoculated in it is a submerged fermenter. So the spores have been inoculated. Once the inoculation is over, then it should be incubated at 30 degrees centigrade in a rotary shaker, and the BOD incubator should also be used it for. The RPM required for this is 150 RPM. And after 96 hours of growth of your organism, then it will be centrifuged. So the production of our protease will be occur only after 96 hours. So if once it is over, then it will move on to the centrifuge at 4 degrees centigrade. So we will extract the crude enzyme. So the crude enzyme may contain the byproducts and also some of the water moisture content and all. So we require to concentrate our specific product protease. Then we will move to the in downstream processing, the protease is isolated and purified with the help of a solvent called isopropanol, and this isopropanol will be further undergo filtration method, and also it is refrigerated at 4 degrees centigrade. So once this isopropanol have been chilled, then again the isopropanol have been added into the solution, so that the precipitation of only our protease will occur. Once the precipitation is happened, then we will the concentration of our product is done, so we will centrifuge it and we will separate only the precipitates. The precipitate is nothing but our protease enzyme, which will be dissolved in the buffer. So citrate phosphate buffer have been used, and a purified form of enzyme have been obtained from the overall production processing. So this is the downstream processing of when we are using the Rhizopus species. Then we will see what are the optimum condition. So it requires 30 degrees centigrade as temperature. The pH should be 5.5. The incubation time is 96, and the cell should be reaches about 10 power 5 the concentration. So if it have been reached that size, then they will go further into the production fermenter. Protease will have a very vast application. That's why we are going for the industrial production of this protease. As I already mentioned it that if we have many different companies or industries which is producing an enzyme or whatever may be the enzyme, almost 60% of enzymes will be only the protease. So that's why we are going for or looking forward the industrial production of this protease enzyme. These are some of the applications where the protease are being utilized for. One is that biofilm removal, then fibrin clot dissolution, and for nematode biocontrol. Nematode is nothing but a roundworm, so which will be present in the plant. It will start eating all the plants, so that can be as a biopesticides we will use it for, and also for wool biopolishing we are using. Then prion degradation. So prion is like a neurodisorder disease where it will restrict the protein degradation. This will be overcome by it. And also for photogenic industries they are using this protease. In leather industries where they are using it, and also for the food industries they are using it, especially for tenderness of the meat they are using this protease. And for keratin residues. So while processing all the keratins or else extracting the keratins for the beneficials in order to add into some of the cosmetics, they will processing. While doing all these processes, they use this protease. And for contact lens cleaning, they are using it as it have a hydrolysis property. If the protein or else an antigen have been present, it will be cleared out or degraded by using this protease enzyme. And in the detergent industries, we know that the stains can be easily broke down, or some of the proteins or whatever may be the dirt or impurities present in it, it will go and bind to your cloth. That should be broken down by using this protease enzyme. And then last one is that silk degumming. So we know that the silk will be obtained from the silkworm. So while they are isolating that silk, they will have been protected by a gum which will be like a shell type. That should be removed, and only our silk should be taken from it for the textile industries. So for degumming will be nothing but the removal of gums present in order to extract only our silk. So these are the applications of protease enzyme. So that's all about the upstream downstream processing and the uses of proteins, then applications of proteins, and also the overall enzyme, what is enzyme, all these things we have discussed in these videos. Yeah, thank you.